Thank you so much for having me here to talk about whole genome sequencing in our PulseNet foodborne network. So just to give you a little bit of background about foodborne illnesses and the impact on public health. So I don't know in this audience, if you really want to think about it, how many of you have had a foodborne illness in your life? Probably many of you, yeah, and I see a lot of hands up in the audience. About 48 million people in the United States get a foodborne illness every year, and about 500 million people globally get a foodborne illness every year. So what that turns out to be is about 1 in 6 Americans and about 1 in 10 people on average internationally. And these are based off of estimates that were generated about 10 years ago. And sometimes when people get a foodborne illness, it's severe enough to be hospitalized. And globally, this means that millions of people get hospitalized with a foodborne illness, and it can be 250,000 deaths each year due to foodborne illnesses. And this just doesn't impact lives, it also impacts the economy in terms of contaminated produce or contaminated other foodborne disease sources that then get recalled from the market. And overall, we see huge impacts in terms of morbidity, mortality, and on the economy every year. So you may be asking yourselves, how do I know what's causing foodborne illnesses? And I'm going to get into a bit of our foodborne disease surveillance system in the United States. So as I mentioned before, 1 in 6 Americans get ill every year. And of those people who get ill, right, it's about 50 million, a subset of them go to their physician. And if the disease is severe enough, so if they're hospitalized or they're just not resolving their illness, or it's an astute physician, they will collect a sample. So most of the samples that we get into our surveillance system are stool samples or poo, if you're in the UK. So once your sample is collected, it goes on to pathogen identification. So this usually occurs in a diagnostic or clinical lab, and we're looking for Salmonella, Shigella toxin-producing E. coli, Listeria monocytogenes, Campylobacter, Vibrio, Chronobacter, so the common enteric bacterial pathogens we see as part of foodborne disease surveillance. If any of those pathogens are identified, there's a national reporting law where that sample needs to be sent to public health. So within the US, we have public health laboratories in all 50 states and some of our larger counties and cities where they have sequencing capacity. They perform the sequencing, and then that sequence makes it to our PulseNet national database. So PulseNet is our national molecular surveillance system in the USA that's been around since 1996. So we're coming up on a 30-year anniversary pretty soon. And then what happens when that data gets transmitted to our PulseNet national database? And I should also mention that all the sequence data we currently generate, it's Illumina data, that also gets transmitted to NCBI and is on the pathogen detection page. We take a look at all the data to look for close molecular matches. So isolates that are have the same fingerprint. And the assumption here is if you have closely genetically related isolates, that the cases where these isolates came from may have been exposed to the same food source. And once we find those close molecular matches, so we look for 3 or more matches within a time frame, usually 30 to 60 days, we tell our epidemiologists about it, and then the epidemiologists go forth and work to identify what the potential source is. So they do trace back to the potential source of the outbreak. And this is just an idea of how many isolates we receive within PulseNet annually. So this is just a scan since 2006, and every bar here represents a different pathogen. The blue line is actually Salmonella, because we receive tens of thousands of Salmonella samples per year. And then the other bars are most common pathogens we receive. So we started sequencing in PulseNet in about 2013, and we fully transitioned to sequencing in 2019 within our network. And currently we have over 500,000 sequenced isolates as part of our national surveillance system. So every time we identify a potentially ongoing outbreak, we can look back to our sequence data to see if there's any signals there of closely matching sequence data that may have come from a particular source in the past to help inform us as we do our investigations for potential outbreaks. And I mentioned PulseNet USA, but we're also part of a larger network, PulseNet International, where we are split into 8 regions and we work with over 80 countries. And our focus here is really to build capacity, data sharing, to share across countries, because food is a global commodity, potential outbreaks, so we can identify outbreaks sooner and then prevent illness and save lives. And that's something I didn't touch on before, but really the goal of what we do in PulseNet is to identify outbreaks sooner, trace it to the source, remove that product from market so that we can then prevent illness and reduce the impact of these diseases on people's lives. So we've been interested in working with Oxford Nanopore Technologies for a while in both PulseNet USA and our PulseNet International network for a few reasons. Accessibility of the instrumentation, not just domestically, but also when you think about implementing this technology for sequencing in low and middle income countries, cost effectiveness, the speed for analysis, so from sequencing to result, and also generating closed bacterial assemblies. But one of the challenges we had, at least in the past, is the accuracy. And being able to get that strain level SNP information we need to identify potential outbreaks. So with the launch of the Q20 Plus chemistry, we were really excited to revisit Oxford Nanopore Technology and really see if we could use it for our ability to generate those molecular fingerprints we look for to find close genetic matches and use this technology to identify outbreaks. And before I can go into the results, I should step back a little bit and talk about the methods we use to compare genomes. So we use core genome multilocus sequence typing. If you're not familiar with that analysis approach, we focus on genes that are in common, are present in 95% of the isolates that we're analyzing, and generally these genes are genus or species conserved genes, and you'll find it covers about 50 to 80% of the genome. So what does that mean for a bacterial isolate? So for Salmonella, for example, you have about 4,600 genes for Salmonella, less genes than you would have in a human genome. And our core genome is about 2,300 genes. So we use a fraction of the genome that's highly conserved to then go on and identify isolates that are closely genetically related by comparing core genome MLST alleles. So I'm going to go through some of the examples that we first started using ONT data for to understand how to get the technology to work for us to detect potential outbreaks. So a lot of what we work on is both E. coli and Salmonella, and this is an example of a Shigella toxin producing E. coli strain called, that's a 0145 serotype. And we were comparing ONT-generated data to our common PulseNet protocol, which is short-read data. And we used different thresholds for sequencing and calling bases, working with ONT to understand why we're still seeing these differences and the number of allele differences compared to our validated approach. And so we had tried different base calling methods, and really nothing fixed it. So when we look at an outbreak and we're detecting an outbreak for 0145, we expect to see 5 or less allele differences. And for this particular isolate, that was part of the outbreak. So the orange isolate on the screen and that light blue circled isolate are the same strain, sequenced 2 different ways. And really, you want that light blue isolate to be 5 or less allele differences from what was identified as part of the outbreak. So currently, looking at this data, you couldn't use it as part of outbreak detection because we were seeing so many differences. So we would have essentially false negatives, right? We would have things characterized as outside of an outbreak when really they were part of an outbreak. And we looked further in the data, and working at CDC in the enteric disease laboratory branch, we have experts that have worked on our different enteric bacterial pathogens for decades. And thankfully, we can tap into them to try to understand what's going on when we see these differences. So going deeper into the data, and what we were seeing is base changes that were associated with restriction enzymes and other potential modifications in the genome. And that was just like a light bulb moment for us to say, hey, there's other modifications here that are leading to this base call that's different than the canonical base for that location. And we worked closely with ONT to communicate with them what we're seeing along the way, and we were really excited about the Dorado base caller. And with the first bacterial-aware base calling, we were able to go back and then repeat that base calling using Dorado, that's bacterial-aware for methylation, and now we see 1 to 2 allele differences. So now we're going from a technology that we couldn't use as part of our outbreak detection to placing that isolate right within our outbreak cluster. So this is really exciting for us, and we went on to do further validations. So this is just an example here looking at a heat map where red is the most similar, green is the least similar, and we have our outbreak cluster and our closely related outgroups as part of this graph. Those entries in bold are the ones that we sequenced using ONT compared to the technology we more routinely use currently, which is Illumina. And you can see, even with those biological replicates, we placed that strain, once again, right within the outbreak cluster. We saw maybe 1 to 2 allele differences, but like I mentioned, that's still acceptable, kind of within the range of what we look at for outbreak detection. And those samples that we sequenced by ONT that weren't part of the outbreak originally also continue to be outside or part of that outgroup, but not part of the outbreak cluster. So it was looking really promising for us. And then we also wanted to go back and take a look to see, okay, so now that we're using this bacterial methylation base caller, how does it affect those samples that we didn't have the methylation issues with, and make sure that it doesn't remove those isolates from the outbreak cluster? And our assumption was correct, that the samples remain comparable to the currently validated method for Illumina. So now we decided to go back and look at more outbreaks to see, are we seeing this issue with other serotypes of Salmonella, E. coli? Is there anything we're missing along the way? And as part of what we do for validation, we sequence hundreds of samples. Like I mentioned, PulseNet has been around for almost 30 years. We have a very large isolate collection at CDC. We like to think about it as a national archive of enteric bacterial samples that include outbreak and also diverse data sets. So we went back to start doing further validations of outbreaks, looking at both isolates that are part of the outbreak and, again, those closely related outgroups. Looking at heat maps, like I mentioned before, seeing how these isolates get placed as we do this sequencing, comparing to our current gold standard method, and looking at different outbreak sources. So previously I was showing you outbreak sources associated with food, and here's an example of transmission that's person to person associated with a daycare facility. And once again, we see that the sequence data generated by ONT matches closely with what we see from short-read sequencing and places those isolates within the outbreak. So once again, we're seeing the validation of data to basically kind of SNP or variant level accuracy for our ability to detect outbreaks and place isolates within an outbreak that are sequenced by ONT. And so really here I'm just showing you some examples of other outbreaks as we went through and did this sequencing, all looking really positive. Here's an example of another E. coli outbreak. It's serotype 0111. And once again, those dark or bolded samples sit nicely with the other outbreak isolates. So overall, it's looking really promising to be able to use ONT sequence data for our ability to detect, for our surveillance system, using it for detection of potential outbreaks. But we ran into some bumps along the way, too. So this is an example of Salmonella Oranienburg. So Salmonella, we talked about E. coli. E. coli has several hundred serotypes, and we're kind of going through different common serotypes we see in doing these outbreak sequencing data sets and validating the technology for our use. For Salmonella has over 2,500 different serotypes. We see there's a common 50 to 100 that we see as part of outbreaks in the United States, and we're going through and doing this validation as well. And so this is an outbreak linked to eggs served in restaurants. And you can see the bolded isolates for ONT at the bottom all have over 80 allele differences that if we were just using the sequence technology alone would have placed them outside of the outbreak. So we've run into a few bumps along the way for identifying additional potential methylations. And here's another example of that. 0103H2, this is associated with a fast casual restaurant. And once again, you see the ONT data kind of being clustered on their own, where you see over 100 allele differences of the ONT-generated data versus the short-read generated data in terms of being able to identify potential outbreaks. So we looked further into this data, and not surprisingly, based on what we'd seen before, we saw additional restriction enzyme modification sites where we see additional methylations. And we really see this as work that we have to do initially to identify these potential methylation sites, and Oxford Nanopore has been really helpful in working with us and improving the awareness of the Dorado base caller to these methylation sites. And I assume we'll hit a saturation point where we've detected the majority of them, and it's really then we can go on and use that data as part of our PulseNet surveillance system now that we're aware of these potential modifications as part of the different enteric bacteria we investigate. So currently we're doing validation data sets, testing several hundred isolates to look at diversity of strains as well as outbreaks to identify additional potential methylation sites to improve the base caller. But so far, technology has been really great, especially new flow cell, to get highly accurate data for bacteria. And we also are verifying antimicrobial resistance gene prediction to make sure that what we see phenotypically from our short-read sequence data, now from ONT, is all congruent. As I mentioned before, we're continuing to explore those additional methylation motifs, doing QC benchmarking. So a big question we get as part of our network is what's the minimum threshold for coverage for bacteria to be able to analyze that data as part of an outbreak analysis. So that's something we're working on right now. And we'll be coming out with validated QC benchmarking or thresholds for when we accept data into our system. We're also in the process of SOP development and looking forward to doing trainings as part of our PulseNet USA as well as our PulseNet International network using the ONT technology. And we're actually doing a training coming up in about a month in Thailand using this technology with our PulseNet Asia Pacific partners. Our focus here is on AMR detection and characterization, but we hope to develop the training further so that we can do this strain-level typing for outbreak detection. And I really want to thank the people within my team and our collaborators that really made this work possible, including Peyton, Justin, Ryan, Jess, and Lee from my group at the Enteric Disease Laboratory branch, plus our collaborators at Doherty, Theogen Genomics, and APHL. And here's just some more information if you ever want to learn more about PulseNet, and a few links there. So thank you all very much.